Okay, I know how you feel, but this is it. And so I wanted to invite you again and all your friends and all your enemies on the next year series. We will have a wonderful, wonderful series of, again, great speakers coming in. So just to remember, Friday, 8:30, if you are interested in vision research, that's the place to be. And again, please share your link with anyone you think could attend and enjoy that seminar. So it will be just fine. Just a short introduction. There is a lot of wonderful things coming from Italy. And really, this is a fantastic place to be from. And many of the great people in science came to United States. And really, my favorite is Loredana Quadro. And she is a great retinoid chemist working in Rutgers and make a major contribution. And another contribution of her that she also brought Filippo with her. So this is fantastic news because Filippo is one of the best structural biologists in our field. And with this introduction, let me go to John. John?
Yeah, I can take it from here. Today is an honor to introduce Dr. Filippo Mancia, Associate Professor and Co-Director of Graduate Studies, Department of Physiology and Cellular Biophysics at Columbia University. Dr. Mancia is a renowned structural biologist with experience in X-ray crystallography and particle cryo-electron microscopy. Dr. Mancia received his PhD at the University of Cambridge in the lab of molecular biology, and he went on to do his postdoctoral training at Columbia University. Dr. Mancia's main research is on membrane protein lipid interactions and high resolution structure enzymes. Example of Dr. Mancia's most recent high impact papers include the Cryo-EM structure of the human ferritin transferrin receptor complex and the structure and drug resistance of Plasmodium transporter PF-CRT. Join me in welcoming Dr. Mancia, who will be presenting a talk about the mechanisms underlying cellular uptake of vitamin A.
Well, John, thank you so much for your kind introduction, and I'd like to a wonderful introduction. I'd like to thank Grazina and for the invitation, and and Chris too for his kind introduction and invitation. I resisted this. I must be honest, and I tried to schedule it as late as I could in the hope that I could actually be there in person, which is something I would have liked to. And I've just made a little deal behind the scenes with Chris, saying that if he even modestly likes this talk, I'm doing it, but I'm coming back. So I'm coming back for real. So this is actually my first, I confess, it's my first Zoom talk. So I apologize if it's going to be a little bit of a disaster. I gave a biochemistry lecture for an hour and a half in about late September on Zoom for the graduate students. And about an hour into it, I lost focus and I started really thinking of myself, people, you've been talking in front of a computer, no one's listening. And I started to laugh at myself, and I lost complete track. But that was after an hour. So I hope I can keep the focus and maybe entertain you a little bit. So the plan of the day, I'll share the screen in a sec, is as follows. I'm going to tell you for about 2/3 of the talk, I'll tell you the STRA6 story. For those, it has aged a bit because it's a story of 2016. But I thought it was nice to give the introduction and to take you through the historical perspective, how we got into it, and what the initial structure told us, and what the questions were that it raised. And then I'll move on for the last third of the talk, so very recent, completely unpublished data on other structural biology endeavors in the STRA6 and results. These are very fresh and very under-analyzed. So I'm not going to give you a conclusive story. I'll just tell you, I'll see this as a research update. Anyhow, it is a great pleasure, in quotes, to be there. It's bloody freezing in New York. It snowed. My Mini is under about a couple of feet of snow. It's not even the electric one, Chris. So, but we have to upgrade on. We have to work on that. Anyhow, so let me just project myself into California mode, which something I love to do, and off we go. I assume that you can hear me. I'm not seeing any complaints.
Yes.
I'm not hearing any complaints.
Yes.
So I'm going to go into presentation mode. And I'm going to get rid of myself. I'm just going to iconize this little thing. So if there is an issue, just speak up, please. Okay, so as John said, I'm a structural biologist, and we've had a long-standing interest in membrane protein and using structure to understand function. And the niche, the biophysics niche, I try to focus on. There's really two. One is mechanism of drug resistance. That links to the recent work on malaria and on TB, our recent papers of 2019, 2020. But we've also had an interest, a long-standing interest, as Chris correctly points out, in vitamin A, in this hydrophobic molecule, and how it gets into the cell, and what the role of, if any, of a specific uptake protein plays in the process. What Chris didn't mention, and he might not even know, is that when I was an undergrad, at this point, many years ago, in the little town of Pavia, south of Italy, I went for a thesis in chemistry, and I thought that crystallography was fun and cool, and I crystallized, I think, a retinol. I'm actually sure an odorant binding protein and a retinol binding protein. I actually did the first purification of the RBP in complex with TTR. So that, in a way, set the stage for future work. So let's see if I can advise. So as all of you know, and this is certainly not the forum to spend too much time on vitamin A. Vitamin A is an essential nutrient. It is so because of its dominant role in the visual cycle, but it is also it plays a role as an activator of transcription in any process which involves cellular differentiation. It is, of course, crucial during reproduction and the whole embryogenesis, in the whole maintenance of the placenta, but it is also essential in nervous system, immune system, and many diverse biological cellular functions. So retinol, vitamin A, typically refers to the form retinol, which is the transport form, which has no apparent biological activity per se. Retinol is the alcohol form of vitamin A. Vitamin A is an extremely hydrophobic molecule, and as you all know, once the retinol enters the cell, it can really suffer two fates. It can either be stored, converted to the ester form and for storage, or it can be enzymatically converted to the beloved retinol, which, as we know, has its role in the visual cycle, or it can again progress from retinol to the retinoic acid, which serves as the regulator, which binds to specific receptors and serves as the regulator of transcription for hundreds, if not thousands, of target genes. We mammals are unable to synthesize vitamin A de novo, so it must be acquired from the diet. And once in the diet, so you eat it either in the form of retinol esters from meat, or from vegetables in the form of essentially two molecules of retinol beta carotene. Once inside, it gets packaged into chylomicron remnant, and it goes into the circulation. From the circulation, it can be the main storage form. It goes into the liver. In the liver, and it's in the liver that it is stored, and it is in the liver that once it's been ingested, and it gets to, in the times of dietary insufficiency, or when there is no dietary supply of vitamin A, it is from the liver that it has to go out. And the way that it exits, it's bound to its sole specific carrier called retinol binding protein, shown here on the right. So once retinol associates with RBP, it exits the liver and it reaches the target cell, raising the question of how does this hydrophobic molecule that needs the carrier, needs the retinol, it needs the carrier in order to be soluble in the bloodstream, can actually enter the membrane. This has been a debate that has lasted in the retinol field, in the retinoid vitamin A field, for literally decades. There are those that argued for and argued against the existence of a specific receptor for retinol-bound to retinol-bound binding protein. The question was to a certain extent set to rest in the question of the existence of a specific receptor for the RBP-retinol complex, and of something that could mediate the transfer of retinol, the union bidirectional transfer of retinol from the bloodstream into the cytosol of a cell, where retinol is known to associate with, in quotes, the equivalent of retinol binding protein outside, which is called cellular retinol binding protein, or CRBP, was set to rest in 2007. And the group of Hui Sun at UCLA, using some extremely clever cross-linking biochemistry, were able to identify a previously known gene to be retinol regulated, STRA6, as the receptor for the retinol binding protein retinol complex. We flash forward 10 years, and mapping of mutations in humans, which are linked to a syndrome called Matthew-Wood's syndrome, which has not surprisingly, a major impact on the visual cycle, and work in mice, again, showing most often ocular phenotypes followed. And what is STRA6? So STRA6 is a protein with 9 at the time, and we're doing a historical perspective here, a protein with 9 to 11 predicted transmembrane helixes, and with no resemblance, no apparent obvious topological resemblance to any other known membrane protein, and certainly no one of known structure. We flash forward another few years, and some again clever biochemistry, mostly from Hui Sun's group, were able to identify the RBP, the region between the transmembrane 6, the predicted transmembrane 6 and 7 for the binding of retinol binding protein, and more debatable regions in the protein, in the intracellular side of the protein, which could have to do with the site of interaction with cellular retinol binding protein. There will have also been reports that STRA6 might not just be a molecule for the internalization of retinol from RBP, but it could also have a signaling role, and it could also be exchanger, i.e. that the movement of retinol be bidirectional. But what came evident at this stage was the novelty of the system, not just regarding the intake of vitamin A, mostly in the parts of the human body, that most need it. For example, in the eye and during development, that's where the STRA6 is mostly expressed, but also from a biophysical perspective. You know, we've seen, and structural biology in this, I think, in my opinion, has contributed tremendously. We've seen all sorts of ways to get ions and solutes in and out of the cells, and let it be by primary active transport with the hydrolysis of ATP, or using electrochemical gradients like in channels and transporter. But it seemed clear from the onset that the way that a hydrophobic molecule linked to a specific soluble carrier, retinol binding protein, on one side, an intracellular carrier, cellular retinol binding protein on the other, the bilayer, and this molecule in blue here, STRA6, how they work seemed one novel, and two absolutely fascinating from the energetics perspective, from the biophysical. How do you deal with the hydrophobic retinol? So we got into this. This is when we got into this, and this was a while back. And I'll just go quite briefly on this. And we performed what we know how to do best at the time, is a structural genomics approach. So we went around and looked for as many STRA6s as we could find, hoping that we would find one that would be amenable to structural studies. And in our first screen, I think we had about 10 of them, we immediately noticed that the STRA6 from zebrafish was by far the best expressed and the most stable in detergent. What I'm showing here on the right is an elution profile from size exclusion chromatography. What I'm showing here on the left is a functional characterization of zebrafish STRA6 in transiently transfected human embryonic kidney cells, showing that the uptake of retinol is actually STRA6, it's zebrafish STRA6 dependent, i.e. proving that the STRA6 we had inferred from a bioinformatics analysis is actually a bona fide STRA6. And then we went on and we spent a lot of time optimizing doing the things that we enjoy doing, which is optimizing the detergent conditions and slowly, progressively increasing, and you can show here on the graph on the left, the stability, the thermal stability of the protein, knowing that this is an approximate correlate to the likelihood of obtaining ordered crystals. But there was always a problem. However we purified it, however well we washed them, however we treated those membranes and added detergent, we would always get, the gels were never clean. And we would always get what we thought at the time was a contaminant, a tightly associated contaminant, which mass spec experiment readily revealed to be calmodulin. And we gave the presence of this contaminant calmodulin, we gave it in part the blame of the fact that yes, we could obtain crystals, but the crystals never diffracted despite extensive optimization, generation of monoclonal antibody fragments, and at least a couple of years of work. Never went really beyond 10 angstrom, despite showing a very pretty pattern just behind the beam stop. This is taken at the NCCAT beamline at APS, one of the best beamlines in the world, and we did our best with these pretty little crystals. However, a dynamic light scattering experiment coupled to a refractive index measurement, i.e. a way to couple the size exclusion chromatography runs with an estimate of the mass across an estimate of the mass and a decoupling of the protein mass with that of the detergent mass, showed us that the main species we were purifying was actually 180 kilodalton, consistent with 2 STRA6 molecules, each is 75 kilodalton, and 2 calmodulin molecules, each is about 15. 75, 75, 150, 15 times 230, 150, plus 30, 180. This observation immediately put the project, and we're talking a few years ago, in the realm of what was technically achievable by single particle Cryo-EM, and in a story which has now become, in the last few years, has now almost become routine. These proteins that crystallized poorly or didn't crystallize at all, where researchers had spent a long time optimizing the biochemistry and the sample preparation, all of a sudden the structural biologist was illuminated because you didn't need ordered crystals anymore. It was still a learning technique. It didn't come easy. I still personally have no clue what I'm doing in single particle work, but I have people who work with me who have learned it, picked it up much better than I have, and are wonderful. And so, to cut a long story short, we jumped on Joachim's camp, and I apologize that this is horrible. This line is crooked. I don't know what happened here today. Anyhow, we jumped on a 300 microscope. You could see beautiful little particles there, and all of a sudden, from the 2D class averages, we could make, without imposing two-fold symmetry, we knew it's a dimer, we could see this UFO-like shape, which with these 2 legs. Now, the 2 legs were crucial for us because, given the small size of the particles, they allowed us to do it. It was key. It was key for the alignments. Flash forward from 2D and the initial model, we were able to go into the 3D classification and generate the density map at about 3.9 angstrom. Actually, good quality 3.9. We've since improved it to about 3.5, 3.6 in. So the one thing I didn't mention is this protein ultimately was a protein purified in a maltoside detergent with cholesterol hemisuccinate, a typical cocktail for the eukaryotic membrane proteins, and was then shifted to these amphipathic polymers called amphipols for the image analysis. So we could obtain a density map. We could build a model, and this is what the first structure of zebrafish STRA6 looks like. So it's a dimer. We knew that. 2, it has 9 transmembrane helices and 1 intramembrane helix. I'll just take you quickly through the fold, but I think it's interesting. It has an N-terminal bundle shown here in blue on the right, comprising the first 5 transmembrane helices. Protein, just through a long loop, goes into these extended helices 6 and 7, which span up to 25 angstroms above the putative plane of the membrane. The chain after transmembrane 7, and you're going in yellow here, after a loop, the chain goes at a helix 8 at a slant, takes the protein through also from one protomer on the right to the second protomer on the left, transmembrane 8. Transmembrane 9 is essentially part of the second protomer, so there is a transmembrane exchange here, and then before coming back through a helix, which becomes 2 helices given the twofold symmetry, that traverse about mid-bilayer and essentially parallel to the plane of the membrane, before exiting the membrane and going into 3 helical segments, which are the binding site for the calmodulin. So calmodulin is, we noticed, is by no means a contaminant, the contaminant that we feared. It's actually what ultimately allowed us to determine the structure of this molecule. And I'll obviously talk more about the STRA6 calmodulin interaction in a little bit. The other interesting feature is that the residues known to interact with retinol binding protein lie at the very top of these long helices that protrude, as I said, 25 angstroms above the plane of the membrane. This down here is a view as a slice taken mid-membrane and flipped, and you can see these 2 parallel, twofold symmetry-related intramembrane helices in orange, and this is just very slanted transmembrane 8, and this is the domain-swapped transmembrane 9, and you can see here the helical bundle. So this is...
If I can just quickly ask you and to tell you that we are live and listening to your presentation.
That's always good to know, Chris.
So this transmembrane helix that runs along the membranes, that's very unusual in protein structure in general because of the dipole issue, right? So you have each helix has a dipole. Mm-hmm. And is that dipole compensated by some side chain like a counterion for it, or?
So it's in a way, it's also shielded within the protein. I'll get to that in a second. But it's not just abandoned by itself, because you have what's unclear from this slide is this part underneath here, which is actually non-hydrophobic. So there is a hydrophobicity on top, hydrophobicity on the side, but one side of the helix can actually be non-hydrophobic, which is down here.
All right.
It's quite interesting. It's actually quite interesting. And yes, the sequence are not the standard TM sequences in which you use the dipole information also for prediction. It was a completely, that's why also you can interpret, you can explain the 9 to 11 question mark on the topology of this thing in the prediction. Have I answered your question, Chris?
Perfectly well.
Okay, so let's continue.
Can I comment something maybe unusual, but I want to say I'm listening to it as the best story ever. Like I'm waiting for the crime to unravel, and that's fantastic. Thank you so much.
You're more than welcome. So maybe we should just stop and not let the crime unravel, and then
please continue. Please continue.
I'll see you all back. That's the way to do it. That's how you do the proper sequel. No, well, I will disappoint you because we don't have a complete story, but that's okay. I'll keep the suspense, and let's keep going. So here we got these weird intramembrane, long intramembrane helices. We've got the yellow and the orange. So what is it? The yellow and the dark yellow, the yellow and the gold, which are all crooked within the membrane. We've got an unclear N-terminal bundle. And essentially, we can define a 3-domain protein. N-terminal, first 5 helixes, central domain, which is massive and imposing in the way that it spans from one side to the other and above. At its apex, there's the known retinal site for binding for retinal binding protein. And then there is the C-terminal domain, which constitute the legs, which and comprise and are dominated by a tight interaction and a complex interaction at this point. We'll define the concept of tightness in a bit with calmodulin. So what is interesting about this molecule? There's several things, but there's really 2 things in particular. The first has to do with the central domain. So a cavity search, if you do a cavity search of the molecule, you see that the central domain is essentially, there is void in there. The molecule is empty from the 25 angstrom of the retinol binding peptide up here to about mid bilayer. So the ceiling of the cavity is the retinol binding protein binding site, and the floor of this massive cavity are the 2 intramembrane helices. So we know, so this spans all the way to mid bilayer, and we know that that nature doesn't like vacuum. So the first question, I'll answer that in the next slide, is really what's inside this cavity. But the other striking feature of this cavity is that in a sort of window, what we call a window, created by the intramembrane helices on its floor, on the base, transmembrane 8 and transmembrane 9, this massive cavity has an opening directly into the bilayer. This is a view from the bilayer. This is a view from above. Obviously, at this point, something that is open to the bilayer in such way is more likely to be hydrophobic than hydrophilic. And indeed, a hydrophobicity plot of STRA6 shows that within the cavity, shown here, this is a view for the top, it is essentially as hydrophobic as the transmembrane region itself, which is consistent with its being open to the membrane. Inside, we find several ordered, partially ordered or ordered lipid molecules, two in particular, always there, bound to a conserved asparagine, a conserved threonine along the intramembrane helices that we interpreted as a tightly bound cholesterol. This is baculovirus, this is insect cell material in which that has not seen retinol. So that's the striking feature one. We've got this massive central domain, massive hydrophobic cavity with an opening to the window. If we take the structure and map the mutations which are known, which have been, thanks to mainly to Sun studies over the last more 10 to 15 years that are known to impact function, it is not surprising that they cluster around the site known for the interaction with retinol binding protein at the apex of this cavity and along the two long transmembrane helices, 6 and 7, that essentially connect the calmodulin binding region, C-terminal domain, to the binding site of retinol binding protein. This information, combined with the conservation analysis, which shows that conservation is clustered in the N-terminal domains of unknown function here, but mostly in the center, in the lining of this hydrophobic central cavity, and in the region that interacts with calmodulin, allow us to do two things. One is to start to think about how retinol binding protein might interact with STRA6. And what I'm showing here is mutants in STRA6, which are known to disrupt the binding of retinol binding protein, and the region shown in yellow of retinol binding protein that is known to interact with STRA6. So we can start building or thinking about the association of the two, and we can then formulate a model of how this whole thing might work. So we hypothesized at the time that interaction of retinol-loaded retinol binding protein with its binding site on STRA6 triggers the release of the hydrophobic retinol into this energetically favorable environment of the central hydrophobic cavity of STRA6 for the retinol then to diffuse out through the lateral opening of STRA6 into the membrane, and then directly into the bilayer for exchange for uptake by cellular retinol binding protein. Consistent with this membrane STRA6 and membrane mediated uptake is the fact that uptake of retinol can occur without the STRA6. That accumulation of retinol within the membrane in a STRA6 dependent manner does not necessarily can occur in the absence of the cellular retinol binding protein, and the fact that you can force other retinoids, which are other retinoids in the retinol binding protein, and that the process still works. In particular, you can actually do the experiment with retinoic acid, which has its own specific carrier, which is not CRBP, and the thing, although less efficiently, still works, arguing against, to some extent, the existence of a direct interaction of a direct and required interaction of cellular retinol binding protein by and STRA6. This is this is our model. This became our model of how things happen up to here. But we've forgotten the little elephant in the room, which is the interaction with calmodulin. I'm just doing an Alex Katz-like representation of this, showing the size of this lateral window, which is more than sufficient for retinol to go in and out. Let's now spend a few slides and focus on what happens at the intracellular side. So 3 segments shown in orange, in yellow, orange, and red. One in particular, the one in red, are the main determinant of this tight interaction of the zebrafish STRA6 with calmodulin. Now remember, we thought this was a contaminant. Whatever we did biochemically, we did high salt, low salt, high pH, low pH, whatever we did, we could not get rid of. We even made truncations in the tail of STRA6. And those, obviously, we had no clue what we were doing. We were taking a crystallographic approach to eliminate what was probably any perturbations of the tail led to a complete loss of a function, and no, to a loss of expression. So it's obviously an important interactions. How do they interact? Well, most of the time when we think of canonical calmodulin, so calmodulin is a calcium binding protein. Let's call it broadly a calcium sensor within the cell. It's known to interact with a lot of membrane proteins and regulate and add a calcium dependency to their activity. I regulate them in a calcium dependent manner. Calmodulin has 2 lobes, has an N lobe and a C lobe. Each lobe binds to 2 calcium atoms and 2 calcium ions. And what we're used to seeing is the fact that the N and the C come together around single helical peptides. What we see in STRA6 is actually completely different. And we see that the 2 lobes, the N and the C lobe, are flipped one into the other, that there is what's seemingly a more loose interaction of 2 peptides from one lobe, and there is the second peptide, the calmodulin binding peptide 2 shown here in red, which fits into the groove of one lobe, but actually makes contacts with the back of the other. So, first, we're at 3.9 angstrom resolutions. Okay, that the phases are good and the maps are great, but have we completely screwed it up? Question 1. So, is there a precedent somewhere? And there is this calcium binding protein, not a calmodulin, from this weird organism, the Cryptosporidium parvum. If we overlay our structure with that, you can see that there are extreme similarities. The only major difference is that the segment shown here in red, in one case for STRA6, in our complex, it's STRA6. In the case of this structure, it's actually the N-terminus of the structure itself. This canonical way of interacting comes from the MLCK CAM complex, which is the gold standard. So the way that it interacts is, in quotes, structurally weird. Not surprisingly, I mean, calmodulin, there's no even point going into the conservation status of calmodulin. Calmodulin is conserved at 99%, which is also the reason why a zebrafish protein expressed in insect cells could pick up an endogenous insect cell's calmodulin. It's always conserved. But also the residues within STRA6 that interact with calmodulin in all 3 helical segments are absolutely conserved, as is shown here. Second question, and obvious that the resolution can't answer, but comparison of our structure of calmodulin with structures of calmodulin with and without calcium show, and then in this spaghetti-like figure down here, show that our structure here is actually consistent with a calcium-loaded calmodulin. So, what does this mean? How tight is the interaction? Can the calmodulin come off? Is there a correlation? Have we pointed to a possible correlation between retinol metabolism and calcium homeostasis in the cell? I don't have an answer to the question, but let me give you a little bit of data. The interaction is extremely tight. So you can see that it's a sub-nanomolar interaction consistent with the fact, especially of the peptide 2, consistent with the fact that we couldn't get rid of it, whatever we did. We solved the crystal structure. We were able to solve the crystal structure of CAM just with the calmodulin binding peptide 2 of STRA6. Suggests, I mean, consistent with the 2 are overlaid here, one in the crystal structure in orange and our original structure in blue, showing that it just takes this calmodulin binding peptide 2 actually to make it, to make the conformational change in calmodulin of the flipping of the 2 lobes. We then went in vivo and we showed, well, we went in the insect cells and then in membranes from zebrafish, and we showed that if we pull down with one, we could blot in a specific way for the other, indicating that the calmodulin STRA6 interaction happens in the insect cells with the calmodulin, with the insect cells from calmodulin insect cells, but most of all happens in a physiological context. So it's a physiologically relevant tight interaction in zebrafish. What does it mean? Work with Sun, subsequent work with Sun. So we reached out to Hui Sun at some point, and around the time of publication, we started collaborating with him. He's the expert on this, and he apparently had unpublished proteomics data of pull downs with STRA6, in which the top candidate that showed up was always calmodulin. So he was not he was ecstatic, but not surprised of the fact that we could see this interaction. And he had data which have been recently co-published with us in a 2020 PNAS paper that starts to show a correlation between, at least in the mammalian STRA6, this is work done on bovine, a correlation between intracellular calcium levels and retinol uptake as shown here. Essentially, there is an inverse correlation. Decrease in intracellular calcium leads to an uptake in retinol. And the easiest graph here is on the right. BAPTER is a calcium chelator. It's a membrane permeable calcium chelator. You treat the cells with BAPTER. You decrease the levels of intracellular calcium. The amount of retinol measured in the form of the retinol ester, and this is actually done in RPE cells. In the RPE cells goes up. This is also correlated, at least in Sun's thinking, and I'm not going to quite go there quite yet because I don't think we're ready to answer at a mechanistic level what's going on, correlates with a shift in affinity of STRA6 between retinol-loaded retinol binding protein and retinol-absent retinol binding protein, suggesting that whether this is the uptake or the efflux mechanism of STRA6 might be correlated to fluctuations of intracellular calcium levels. However, when we used our Zebrafish STRA6, so wait, hold on a second. So this is the analysis of calcium versus STRA6. If we now go and look at the presence of calmodulin associated, and this is bovine STRA6, Sun showed rather unambiguously that STRA6 is associated with calmodulin in both culture cells, in both, what is that? I think he did COS, he did 293s, and the RPE cells as shown here. But he also showed, and we should look here, that the association of the two requires calcium, i.e. the chelation of calcium leads to a dissociation of calmodulin. While in our case with zebrafish, we were unable to note in our functional assays these differences, and there is obviously the possibility that we are, we didn't do the experiments properly. I don't exclude that, but there was the obvious observation that whatever we did, unlike what we're seeing here, we could not get rid of the calmodulin. So this, in a way, can be explained by in vitro measurements of affinity between calmodulin and the STRA6 peptides, and you can see that this is the fold difference between zebrafish and bovine. You could see that the association of calmodulin in the zebrafish context is way tighter than in the bovine, in the mammalian case. So it is obvious that the old STRA6s share this association of calmodulin. It is plausible that the calcium sensor aspect of it we haven't either been able to identify to nail it, or that the fish and the mammals behave in a different way. So here we stood. We have two major questions to answer, and one is which are all related on how does this molecule work. So given the fact that we had an initial structure of a non-mammalian STRA6, and the structure was in an amphipathic peptide called an amphipol. If we want to use structural biology to study how retinol, that is hydrophobic molecule, goes from the outside to the inside of the cell, it became obvious to us that we needed, first of all, to shift from the amphipol to a media which was more similar to that of an endogenous lipid bilayer. So to cut a long story short, that's one. And the second thing is that we needed a way to reconstitute a system. So that the first part came rather easily, and we set up and we have a liposome-based retinol uptake assay that works, so we can actually control what goes into the liposome, what's outside. And this is a very precious tool in studying the function of this, and it shows the mini little scheme gel of the purification of liposomes with incorporated STRA6, and the effects in terms of retinol accumulation of treating the liposomes with holo-RBP. But from a structural biology perspective, we decided to pursue the nanodisc route, i.e. to reinsert STRA6 in an environment which is as close as it gets to that of a native bilayer. I'll cut you the technicalities of the detergents we were using and the agony of getting rid of some of these and the reconstitution in the nanodisc, but we were ultimately able to sort out the biochemistry, and we have solved the structure of zebrafish STRA6, which now goes to about 3 angstrom resolution. We've refined it since. Shown here, the red is the nanodisc, showed here, inserted in the nanodisc, and you can see here the density, the density map. I particularly like this image on the right, where it shows the STRA6 model in the dimer in yellow, the nanodisc scaffold shown here just in blue. And what does this structure tell us? Well, first of all, it starts telling us how the protein interacts with the lipid. You can see that surrounding the molecule and within the central cavity, there's all these all these densities, which are which we attribute to lipids, and we could build. So we could build a better structure, and we could start to understand how the protein and the bilayer interact. And there's obviously some conformational changes. Are these physiological? Could it be a minor distortion of the amphipole media in the initial structure? That I don't know. So we adapted biochemical techniques to determine a structure at about 3 angstrom resolution of Zebrafish STRA6 in a nanodisk. But from Sun's data and from our data, we know that there seems to be a fundamental difference in how the two work. So we went at the quest of trying to determine the structure of a mammalian STRA6, and we went back to the drawing board, and we made 2 observations. We used the power of nature, the structural genomics approach of having nature choose which ortholog to work for. So we had 2 extremes here. We had a bovine, we'd worked with bovine, we'd worked with human. We knew bovine would express, but bovine was unstable in detergents. We could never get it to a structural biology-like quality. We also knew that human was a dreadful expressor, and we couldn't get anywhere with it. So we went and we mapped the sequence space between the 2 of them. We thought we tried to go get as far away from human as we could, and lean in a different direction to bovine. I'm representing here in a kind of 1 dimension what's actually a 3-dimensional sequence space. So we picked on this tree, or 2 dimensions. On this tree, we picked size species, and we put a GFP. We screened for expression and monodispersity in size exclusion chromatography, and it became obvious that out of the 8, or what was it, 6 or 8 we screened, the sheep shown here in orange was actually the best. Was actually the best one. You can see here a purification of the sheep from a more preparative purification. You can note that calmodulin there is loyally bound. Go on. We put it into nanodisk. So we immediately went into nanodisk, collected on the Krios. The 2D class averages are shown here. Show the 2 characteristic legs of STRA6, and here is, boom, first time I show this, the structure of a mammalian STRA6 on the right, and the structure of the original zebrafish STRA6 on the left. You can see that at first approximation, they are absolutely identical. But you can start noticing that the separation between the 2 legs, the 2 calmodulin binding segments, is greater here than in the zebrafish. This is now 3.3, 3.2 angstrom. So the overall fold's the same. There are details. And a morph of the 2 structures shows that the changes on this side result are transmitted along these long helices that span from the inside to the outside to the retinol binding protein segment on the top. So if we look more carefully, you can see within the membrane there's a movement of the pentahelical and terminal domain, a movement of the long intramembrane helices, but most of all, there is a movement of the RBP, an apparent movement of the RBP binding region. What this means, this is preliminary and we don't know yet. So we've put it in nanodisk, the zebrafish, and we've solved it. We found a mammalian STRA6 that seems amenable to structural studies. We've solved it too. It is obvious that ultimately we want to have structures of the entire transport cycle. First thing we did, and this is on the zebrafish structure, and again, these are very preliminary data, is we took the nanodisk preparation. This is zebrafish STRA6 inserted in a nanodisk. We treat the nanodisk with retinol bound to retinol binding protein. We wash off the excess retinol binding protein. We measure the accumulation of retinol within the nanodisk. We determine the structure. We look at the differences, and you can see that in a retinol loaded retinol binding, retinol-loaded STRA6, the region of the molecule up at the very top of the central cavity here undergoes a substantial conformational change, shown here, comparison to the apo form minus retinol, and shown shown at the more molecular level of details in stick over here. That's experiment number 1, which obviously, if we'd had conclusive evidence, which we don't have yet, of retinol present inside the central cavity, I would be ecstatic. We're not quite there yet. We're also not quite there with resolution. And then I'm going to close. Let me check my, oh, it's super, I think what are we doing? Oh, yes, it's okay for time, a little bit late. And I'm just going to just show you, close with a little glimpse, which is literally fresh off the pot of today, where we've painstakingly gone towards the reconstitution. What I think is the holy grail of this project in terms of structural biology is to trap the association of STRA6 in complex with the retinol retinol binding protein. And these are, as I say, literally data that I've been given last week and this morning in the next slide. And you can see that there are, if we do a mix, we try to prepare the complex. It's not perfect. You can see that you see classes. You can see on top here, these are the 2 legs. On top, there's nothing, just a little dimple there. But there are also 2D classes which show, look at this one in particular, the presence of something else. And I'm going to leave you with this, you know, just as I said, fresh off the expensive Columbia Krios association of first images of what might hopefully turn out to be the complex of the mammalian STRA6 with retinol binding protein, which we are collecting now. So let me just bring this all together. We've used single particle Cryo-EM to determine the STRA6 is a cell surface receptor for retinol binding protein bound to retinol. We use single particle Cryo-EM, we determine initial structure of zebrafish, show an interesting novel fold with 9 transmembrane helices in this long intramembrane helix, massive hydrophobic central cavity, and a tight association with calmogeny. This massive hydrophobic central cavity led us to make the hypothesis of how STRA6 might mediate the uptake of retinol using the membrane as part of the transfer cycle, and that the whole process, led us to hypothesize that the whole process, given the tight association with calcium-bound CAM, might be in some way regulated by calcium. We aware of the limitations of our initial work because it was zebrafish and not a mammalian STRA, and it was in an amphipole and not in a nanodisk. We were trying to revert this. So we have solved structures in the lipid environment of a nanodisk, both of the zebrafish and more recently of a sheep ortholog that we identified through a second round of structural genomics screening. And we are working towards the completion of the transport cycle. So we have data of the protein which has been loaded, RBP loaded of retinol, and we're working actively on determining the complex with the retinol binding protein. Questions still remain, and I will happy to answer some of them. But let me first of all make the acknowledgements. The initial heroine, Yunting Chen, that was the star biochemist in the zebrafish work, helped by colleague Oliver Clark at Columbia Cryo-EM. Where, this was at the transition, what became our transition from X-ray crystallography to single particle Cryo-EM. The non-published data I presented were spearheaded by Brianna Costabile, former tech of Loredana Quadro, the graduate student in my lab, brilliant Brie, and a lot of the credit for all the sheep work is hers, and the work on the complex is hers. I also would like to acknowledge series of collaborators. You don't move a vitamin A molecule at Columbia without consulting Bill Blainer. You don't move a molecule, vitamin A molecule anyhow without consulting with Loredana Quadro. David Weber, biophysicist at University of Maryland, has helped us with the measuring doing biophysical experiments on the calmodulin STRA6 interaction. And when we needed zebrafish membranes for in vivo experiments, Chiara Manzini, now at Rutgers, was with us. And last but not least, the NEI. So just the last slide is an adaptation of the group in a Zoom joint group meeting with a group of Oliver Clark. I thank you for your attention. I'm happy to take questions.
Thank you, Filippo, so much.
If I can find you, where are you? I'll stop share. There we go.
All right.
Okay, good.
Thank you very much. We have a bunch of questions. We'll start with Dorota.
Hi, this is Dorota Skowronska-Krawczyk. I'm speechless. I mean, this presentation was amazing, and I'm really looking forward for follow-up stories. I will go back a little bit at the beginning of the. Well, apparently I'm not that speechless. I would like to go back to the beginning of the story when you mentioned that there in the initial molecules, in the initial structure, you have discovered cholesterol instead of retinol, obviously because of the use of the system. But I was wondering if there is any competition and if there is a higher cholesterol levels, if there is possibility to compete retinol. I don't know
that's a very good question. So there's a potential problem here is that, as the membrane protein biochemists know, these bloody eukaryotic proteins like the maltosides, but they much prefer maltosides when they get doped 1 to 5 or 1 to 10 with cholesterol hemisuccinate. So the micelle that carries the purification through is 20% a cholesterol derivative. We know that the succinate, the hemisuccinate group hydrolyzed, and both you're left with a cholesterol. So yes, we could build a model on the importance of cholesterol in the interaction, but we could also be a little bit careful due to the presence of the CHS. Now, the second data, which I haven't shown, the second aspect is that if we go look inside the sheep, we see the same densities. If we go look inside the zebrafish with the retinol loaded, we still see the same densities. So what we've done now, and I don't have any results to show because it's a very preliminary, we've had a good collaboration with native mass spec people, a group in Arizona, who's a former postdoc of Dame Carol Robinson in England. And so we're going to use that technique to see if we can see retinol bound and to really probe the tightness and of that interaction.
This is great. And
sorry, there's one last bit, which I obviously didn't show, is that the mutation of the threonine and the asparagine, which apparently make that that interaction with the OH of the cholesterol, you can mutate them. You get a bit of decrease in transport efficiency, but nothing to write on about. So is it a structure? Does it give it the hydrophobic milieu? This thing is lipid. As I see it, you don't even want a high affinity site of any sort in that cavity. You want the retinol in, you want the retinol out. The last thing you need is for it to be stuck there, because then it would defeat the purpose. The other thing is that the competition,
right? So then the competition can be really high, and especially in the liver, where the cholesterol levels are quite substantial.
Exactly.
All right. Let's move on to Aisha.
Well, I can't see Aisha, but I will hear Aisha.
We cannot hear you, Aisha.
Aisha must be muted.
Can you hear me now?
Yes, I can.
Hello?
Aisha, we can hear you.
Can you hear me now? Okay. So my question is, you know how the decrease in on calcium can affect the retinol uptake. I'm wondering, is the opposite is true?
So
when there
wait.
is the
increase. Yes, they're opposite. So I focused on the right panel, in which there was a calcium chelator in the cells. So I add BAPTA, calcium levels goes down, retinol uptake goes through. On the left panel, I added a calcium, I added an inhibitor for removal of calcium, I can't remember, was it Tepsigardin, whatever the hell it's called. So I did exactly the opposite. And I elevate, I mean, I am using the royal we. This is Hui Sun at UCLA. I didn't do anything here. It's way beyond my pay grade, these kind of experiments. And they show that in an elevation, if you put in crude terms, if you pump up the calcium, you get a decrease as opposed to base levels. So the answer to the question is yes. I just didn't focus on the panel of the left of that slide.
And we do not know what the effect of that, right, on cellular function.
No, so I know I can give you an effect on the uptake, on the uptake efficiency. I cannot give you an effect, a more global cellular stress response or whatever, that I can't.
Okay, Alex. Alex, where are you?
I'm right here, Chris. Thank you very much. Sorry, can you hear me okay?
Alex, I can hear you.
Hi, Filippo. Very nice, beautiful talk. Thank you very much. Just a very quick question. So did you see any binding to CRBP from the cytoplasmic side of STRA6? Either
no. So we have not been, we've tried.
Okay.
We've tried and we have not been able to detect. So we've done this, but there is a caveat again to these experiments. So that's why I'm cautious. I'm biased because there isn't an unambiguously identified, bona fide approved cellular retinol binding protein interaction site. The conservation on the intracellular side, aside from the regions that bind CRBP, that bind calmodulin, is poor. We tried it. We tried it on zebrafishes, and we tried it at a stage where we couldn't get neither the RBP nor the CRBP to bind. So yes, we've tried it. We should try it again, and we will try it again now that we have the sheep going. Now the sheep I'm showing, by the way, on the retinol binding protein is after going ballistic on trying with zebrafish to cross-link and liposomes and to resolubilize the liposome, all sorts of ways, is actually mix and freeze. Huh? So, because it's always bothered me that the reported affinities for the two partners, RBP and STRA6, is supposed to be in the nanomolar range. So we should be able to trap a complex, and we have been unable until these very recent. And by the way, just for since we don't take ourselves too serious and we like to be transparent, the last slide I showed was actually from 10:50 this morning, New York time. So we collected overnight on the Krios.
Great, thank you very much.
Yeah. So stay tuned on the CRBP story.
Grażyna and Jim.
All right. Let me see. Okay, so I am following on that CRBP story because there was this slide that you were showing that maybe retinol gets moved out of the STRA6 cavity into the membrane, basically on the cellular side. But one could imagine that this membrane becomes so saturated. Would you expect that actually this could go the other way, that retinol could be transported the other way?
100%, 100%. I mean, I didn't present because it's not my, I presented some data because we co-published them with Hui Sun on the role of, on how calcium, affects uptake. What I didn't present are data showing how levels of calcium impact the affinity for one retinol binding protein, the holo, versus the apo. So the concept that the STRA6 could, if not, maybe its major function is in a situation, for example, in the eye, where you have to offload excess retinol. It's actually as a response to surges. Would it be surges? I think it would be surges of intracellular calcium. Is plausible. Now we're going to try to exploit this in informing the complex, and try to get calcium, retinal status of STRA6 and retinal status of RBP synchronized in order to increase the number of particles that have the association. But it's completely plausible.
Thank you. Beautiful story.
Thank you.
Jim and Grzegorz.
So thank you very much. That was really fun.
Thank you, Jim.
I have a question about the parallel helices in the plane of the membrane.
Okay.
It looked, and of course there's the electrostatic problem, but it looked to me like the molecules, the dimers solved the electrostatic problem by having a parallelogram ring with two short helices, two long helices, and kind of going around in the—
Well, also underneath, in the sense that the underneath part that you're not showing, I mean, it's not, those helices are mid-bilayer, but
yeah.
you have the under part is protein. It's
yes.
not bilayer.
And of course, the mid-bilayer is the place where the dielectric constant is going to be the lowest, and
yes.
you need the greatest electrostatic shielding. So there's sort of a two-part question. One, is there any indication that the plane of that parallelogram could be distorted during the transport process? And have you thought about getting in touch with a tame MD guy, molecular dynamics guy, to see if they can look at possible movements of the protein? Okay,
so the answer to the first question. The first hypothesis, when we looked at the structure at the beginning, we see these things, these long parallel helices. We see these cholesterol molecules, the asparagine and the threonine super conserved. They're making a bond with the cholesterol. We think, thing comes in, opens up, retinol goes through. That was our hypothesis number. Initially we hadn't analyzed it. So when we mutated the TNNN, nothing happens. We found a lateral window, this lateral opening, and we looked underneath let's say not the ceiling, what's it called, the floor under these intramembrane helices, and there's charge, and there is poor conservation. So that in a way distracted us from seeing them as having a major functional role, rightly or wrongly. So we focus more on the hypothesis of the diffusion outside. So we have not, as a consequences, the answer is no. We have not. We have yet to go into MD simulations on this project now. And I'm, I confess, I've kept away until the last about year and a half. I've kept away from MD simulations, and I didn't appreciate them. And I've now have two projects with deep involvement in MD simulations, and I'm loving it. And I'm really I'm beginning to understand, how much they can really give. So that's an excellent idea, and one that we should definitely pursue. And I'd like to bring in more structures to the table, and to see. So we'd use STRA6 as we give it to a group in Cambridge that published methodology on doing MD simulations in lipid bilayers. There was something, but it wasn't, the attention, it was more of movements of the retinol binding protein peptide, and we weren't focused on those helices. That's a great idea.
Thanks.
So I'm personally really excited about your RBP complexes with STRA6. I think that's phenomenal.
Cautious. Let's be cautious on that, huh?
But it's very exciting preliminary data. So I'm going to continue the questions on the intracellular CRBP. And my question has to do with, CRBPs, it has this alpha helical cap on it. Mm-hmm. And that alpha helical cap, there's a lot of studies, from the 90s talking about the dynamics of these alpha helical caps. And, the question that I had, is it possible that the reason that there's no reported affinity for the CRBPs for STRA6 is that there's only affinity for the open form of CRBP, because that's the only form that could actually take ligand on, and that the only way that you could see affinity would be to have a mutant form of CRBP where the alpha helical cap would only be open. I'm curious about your opinion on that.
So your view, so what you're telling me is that there's a possibility that the form, that it's only a very transient interaction due to the fact that there is a discrepancy between the form that cannot take the ligand and the form that we would be measuring affinity for, is what you're saying.
That would be my opinion.
Yeah, that would be mine. But there is always the fact, which has bothered me, that if you go look, okay, there's two aspects of this. The only time we can make a mutant in our hands on the molecule and actually abolish activity, transport activity, is up at the site of interaction for RBP. Anything else we do either has no effect or kills expression.
Okay.
So that, you know, so the fact that we can't pinpoint, and coupled to that is the fact that the conservation on the C-terminal domain, besides the segments that interact with calmodulin, which are 100% conserved along STRA6, is pretty poor conservation plot. Now, does this mean that one of the three, the loosely associated, what I call the zero peptide, could pop off and bind to, and you know, and recruit the CRBP? That's obviously plausible. But the moment that you have this opening of the window, let's think about another angle. The moment you have this opening to the membrane from the protein, you've got a central cavity, retinol goes in, retinol goes out. Do you really need a CRBP binding site? One thing I'm pretty sure of is that the retinol is not going through the protein. It wouldn't make any electrostatic sense, because in the inner part of the leaflet, the electrostatics is simply not there for these intramembrane helices to open up and let a retinol through. And you've still got to do with the fact that you can't preclude the retinol to diffuse if it wants to, because it fits there. Of course, you could imagine something in which you want the CRBP close by, so that you can capture a retinol, a proximal retinol. So that could be, there could be a loose association which recruits CRBP. We have not mapped that, and yes, you could be right in your hypothesis.
No, I mean, your model makes total sense too. I'm just curious, right? I mean,
no, again, you know, we tried and failed. But we tried and failed. We could, when we also failed, and we failed for a long time on the other one, on the more known interaction between the RBP, the extracellular RBP, and the STRA6. So these experiments have to be repeated. And we have by the way, we have all the tools to do so.
Right, we had a wonderful discussion. If there is any last quick question, anybody brave? Dorota, are you satisfied?
I am so, I mean, I'm already screening the literature. I love it. And I would just think about the native surroundings of the STRA6 in the membrane, basically the native composition of lipids, and the dependence of the status of the, for example, liver in the disease or in health. Right. I'm very fascinated by this story. Thank you very much.
Thank you so much.
All right, let's have a victim for the final comments. Who could that be? Anand, maybe not. Maybe Emma, not. How about Professor Kaiser?
Where are you? Oh, so we've got the Cleveland friends here sneaking in. That's great.
Thanks for taking time to present. It was a fantastic story. I was really excited to see the sheep structure and your work with the RBP4 binding. I had two quick questions. One, I noticed you were using human RBP4 with sheep. I was wondering why, if using sheep would be helpful for maybe, you know, having a more native.
I think they're identical, Phil. I think they're identical, and you know, we went with what we have, because really these are we've stalled on this project for a couple of years. And now, boom, all of a sudden, and we don't have, the sheep STRA6 came before the sheep RBP. I do think that they're about 95% identical. And so I'm not worried of the cross-reactivity, but we have to definitely verify that.
Okay. Last thing.
Sorry, Phil. The other thing is that we know that bovine and human, you can mix and match it without problems.
Okay. Cool. In your nanoparticle structure, I know in the original zebrafish structure, you had this lipid that was sort of modeled within the bowl area, you know, where the RBP4 is supposed to bind. I might have missed it. Do you see that in the nanodisk structure as well?
No, the only lipid, if I'm not mistaken, if I remember my own modeling, I'm using the royal we here, is the lipids, we see lipid density within this hydrophobic cavity in any state, let it be nanodisk, no nanodisk, and amphipole, no amphipole, we see that. The only ones we model were the two cholesterols at the smack in the, the twofold symmetry related cholesterols making contact with the threonine and asparagine I've mentioned along the whatever you call it, the parallel helices.
Right.
Filippo, thank you so much. And
Chris,
everybody, happy holidays, and leave you with fantastic music. Listen to that.
Leave me with fantastic music. Thank you so much for taking the time to invite me and to listen to me. I've really appreciated this. Thank you.
Thank you very much. Yes, thanks.